Hi, this is Miss Litton, and we are continuing to review for our APBio exam. Say hey. And so we're still in Big Idea 1, which is all about evolution, but now we are on 1B. Hit the lights. Okay, so 1B, organisms are linked. This sounds like evolution because organisms change over what time? Specifically, what changes over time? Remember population, genetics, we looked at alleles, right? Allele frequency and allele, remember last week? Allele frequency, okay. So organisms are linked by lines of descent from common ancestry. So that's what 1B is all about, is for you to see that all life is linked together. So the first thing we want to talk about is we want to look at the different, what would this be, by the way? What are you looking at here?
Different domains.
Okay, domains. What domains do you see there?
Archaea.
I see bacteria. What else do you see? Eukarya. What's missing? Archaea. Yeah, one of the domains is missing in this picture, right? Archaea, right, of our 3 domains. And so if we're going to look at the attributes of life, the first thing we need to know are the characteristics of life. So let's review that. Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli. They reproduce, they grow and develop, and they adapt and die. Good job. All right, so all instructions for life is in the DNA, and we talked about the central dogma. We talked about the universal genetic code, that the same sequences of nucleotides will code for the same proteins, right? But there are multiple ways. For instance, if you want to get proline, look at all the different ways that you can get proline. So there is some overlap, right? And then we talked about what would this be a good picture of? If I was going to title this picture, what would I title it? Yes, boom shakalaka, the central dogma. And so when we have DNA and we want to make more DNA, what is that called? Replication. When would we want to do DNA replication?
During mitosis.
Or meiosis. Anytime you make more cells, you need to make more code books to go with those cells, right? So during mitosis or meiosis, that is when you would do DNA replication. Then we know using the DNA, look at the numbers. What do those numbers mean? 3' and 5'. What is that referring to? The carbons within the sugar. Right, the carbons within the sugar. So we're using the leading strand, right, as a template when we make our RNA, and that's transcription. How is the RNA different from the DNA?
It has
uracil, wherever you would want to put thymine. What else?
It's
Single-stranded instead of double-stranded. Yeah, that's it. You're saying it. Ribose. You have the sugar ribose instead of deoxyribose. Do you remember that now? Ribose. Super sorry. We don't know exactly where you got left off. Something came and interrupted. So basically what we talked about, this might be a little review. We brainstormed about that not all DNA is a coding sequence. There are non-coding sequences of DNA. So you might pause for a minute and try to brainstorm some non-coding sequences yourself, unless it is actually on this recording. Otherwise, let's tell them now. Pause if you want to think for a minute. What are some examples of non-coding sequences? Stop codons. Introns. rRNA, tRNA. Telomeres. Good. What else? Promoter, operator. Good. Yeah, where transcription factors bind, activators, and enhancers. So the reason why we brought that up is not every bit of the DNA is actually transcribed and translated into a protein. There are other areas as well. So we hope you got that. Super sorry if you didn't. Okay, so now here we're looking at, there are other places besides nuclear DNA, right? Or having a just central chromosome in a bacterial cell or archaean, which would be a single circular, right, chromosome. Where else could you have DNA? We said mitochondria contains DNA. What else? You said chloroplast contains DNA. Give me another one. You're manipulating it even as we speak. Plasmids, extra rings of DNA. So wherever you have DNA, you have the potential to code for proteins. Why is it that mitochondria and chloroplasts have DNA in them?
Because they are originally.
Give me a name. What's it called? Endosymbiotic hypothesis, right? Which says, now tell me.
They're what's free-ranging prokaryotes.
Free living, yes, free-range chickens.
Yeah.
Free living prokaryotes that went to live inside of another eukaryote, right? What other evidence do we have besides that they have their own DNA?
They use ATP.
That, okay, they use ATP. What else? They are self-replicating. They have their own ribosomes for making their own proteins. They're not a single membrane. They are a, which would be indicative of being engulfed, right? Okay, so those are all pieces of evidence for the endosymbiotic hypothesis. So if we look right here, maybe you could pause for a minute because we're going to let people now, maybe the youngest bio buddy that you're sitting next to, give a little explanation of what you see here. And I will pause while you guys are explaining. Okay, so this is a pretty complicated diagram, right? But you understand the basis of it. What it's showing you is it's showing you a nucleus has DNA in it. It's also showing you that a mitochondria has DNA in it, right? It's showing both of you that similar pattern. And here we can see mRNA. Why do they have some A's right back there? What does that mean? It's the poly A tail, right? Everybody gets a cap and a tail. Okay, and some of that DNA is going to be transcribed into a ribosome. And it's showing you the size of the ribosomes. And then it's showing that from that mRNA connecting with a ribosome. And then we also have tRNAs that are transcribed. And then together they work to build a protein. And then you're looking at some of these proteins. The DNA right here is coding for some of the proteins. You know what these are? Yeah. Reductase, cytochrome oxidase. See? You know these. These are the proteins in the mRNA. What do we use those proteins for? Proton, right? Now, yeah, you're moving what? Yeah, across the membrane, right? So you can collect them, right? And then when they come back through, what can you generate? ATP. Okay, so it's just kind of showing you the interconnectedness between them. Now, what you also want to see is that mitochondria cannot exist independently anymore because they've, given up some of their control of what they need to the nucleus. And you can see that in the proteins they're synthesizing right here that are needed for cellular respiration. All right, so I want you to see that because sometimes a picture might seem overwhelming to you or a diagram that they use. Chances are, whatever they use, you've talked about it once. It just may be in a new form. So don't lose confidence. Just go, okay, wait, I'm going to know about anything I see. Let me figure this one out, okay? And look to find what kind of things you recognize. All right, let's take a look at this picture. Glycolysis, who does it?
All cells.
Where do they do it?
In the cytoplasm.
Right, and that probably is the first step. Is this an aerobic or anaerobic process? Anaerobic process. Glycolysis does not require any oxygen. Why? We're not reducing any oxygen with hydrogens, right, and electrons. We're not doing that. So this part is anaerobic. What you have to have the ability to do, though, is once you form that pyruvate, thank you so much. Did it seem like it stopped or ran out of paper?
It stopped. It was at 0.
Okay, yay. Thank you. These are your practice exams. I didn't have time to stay and kill 20,000 more trees, but I didn't want it to get really... What one is this one? Did I do for you? I think this is 2015, but I have the 2016. What? What? Yeah. Okay, so come back to me. So the concern is that in the process of glycolysis, we will make ATP, right? We will net out ATP. But we will also take the electrons that were in the glucose and we'll transfer them to NAD, which means we're reducing that NAD. And we're going to run out of NAD. So we have to have a way to regenerate that. So there's a couple different strategies on how to regenerate it, right? What are those two strategies? Oldest, whose turn is it? What? Oldest, go. What are the two strategies you could do to regenerate that NAD? Fermentation, two strategies in fermentation. Hmm? Yeah? Yeah, lactic acid makes our muscles sore, right? Alcohol. And what you end up doing is the NAD is reduced, so you oxidize it. So you take the pyruvate and you reduce it in either to lactic acid or into alcohol. Mm-hmm. But when you reduce it into alcohol, you generate what? Carbon dioxide. Then by reducing the pyruvate, right? Remember we're here? Okay? By reducing that pyruvate either to lactic acid or alcohol, you end up oxidizing the NAD, so you can do the whole process all over again. But it just doesn't generate that much ATP, so it's better, right, if you use oxygen as the final electron acceptor and you just continue to pass the electrons along, and you can get more energy out of that glucose molecule. Okay, could I please have, oh, and the other thing I wanted to say on that is keep going back to our big idea here. Okay, so look right here. Organisms are linked together by lines of descent from common ancestry. So we can see that commonality in the central dogma. We can see that commonality in glycolysis, right? And then here's another way. If you're going to go outside, let's compare the different types of cells. And I believe it is youngest. Go ahead, compare and contrast. What does prokaryotic include? Bacteria and archaea. What I happen to be showing you is a bacterial cell. This happens to be E. coli, but prokaryotic is under the umbrella, right? I mean, archaea and bacteria underneath the umbrella of prokaryotic. Okay, go ahead. Have these been drawn to size? No. What size should that prokaryotic cell be? Prokaryotic cells should be what? It's much smaller. It would be the size of maybe mitochondria or chloroplasts, right, in a eukaryotic cell. Good job. Okay. You can also contrast an animal cell versus a plant cell. What are some differences between animal cells and plant cells? Plants have a cell wall, large central vacuole. Animals, they have chloroplasts. Yeah, that's a good one we should talk about. What else? How is something they don't have? Yeah, they have to generate trigger pressure. Churros. They don't have centrioles. Good. All right. So here we can see the three domains, and you could easily Google and just review Barchaea. That's a new kind. I combined them. Barchaea, all prokaryotes. Thing we would know about archaea is they are what? Streamophiles. Can you tell me one that, yeah, huh? You knew I was going with that, yeah. All right. So then when we look at phylogeny, okay, we are looking, no reason to stress about phylogeny, right? We're just looking at the evolutionary history of an organism and just plain connect the dots, right? And so we could put everything together and we say that's our ancestral species, right? And then we just start dropping some of them off because everybody else has it, but you don't. Now everybody else has this, but you don't. Everybody else has this, but you don't. What you aren't going to see when you set those up is you have to look out that it's not, the same thing isn't going to evolve twice, more likely. You know, you're not going to see them lose it and then it re-evolves later in the chain. You know, so you have to kind of make sense of all those derived characteristics as you see them. And you look to see, common ancestor for A, B, and C is here. Oh, it's already written on there, sorry. You can see the common ancestors, right? Yes,
So how would you read that in like terms of characteristics? Like would A have dropped off the characteristic, then go to B and C, and then B would have dropped
Yeah,
off?
or maybe A has a different characteristic.
Okay.
This ancestor, though, A, B, and C all have a commonality here. Yeah.
Okay.
Now, it doesn't have to be birds or wings. It could be proteins. It could be the proteins that you find in a sequence. It could be genes that you're comparing. Okay, and that's more likely. Because now that we have biochemistry, right, we can analyze the proteins they contain or the gene sequences they contain, and that's how you're going to draw your phylogenetic tree. And they might say, what do you think is a better phylogenetic tree, A or B? You know, and you would look to see where do you have the most commonality staying together. Which do you think is a more important factor? What would you like to look at if you were going to analyze this? All right, and so when you do a phylogenetic tree, okay, systematics is just the study of the diversity of organisms, and then the phylogeny, like I said, is looking at the evolutionary history. You use systematics to develop your phylogeny. Then here, a taxon. I'm going to step out. Youngest bio buddy, is it your turn?
Sure. Yeah.
Go, Young. I can't keep track of it. You know I can't. Go, Young. Talk about this.
Okay, yeah, this is what I do. A clade is a grouping that includes a common ancestor and multiple descendants.
Can you tell me, why is this not a clade? Two different groups. So we're not going to pull them out like this. And this is not a clade because we're not including him. It has to be along a line. That ancestor and all the derived characteristics are in a clade. You can't jump around with your stuff.
Could it be the whole thing?
It could be the whole thing. You could call this a clade, right? But you can't leave those out, right? So when you look here, this is a great way to see how you're still saying clade every single time, okay? Because it's just the yellow, or we're going to include this whole branch. Now that's the blue, or we're going to include this whole thing. That's a cladogram, right? So we looked at, in cladistics, we were saying, okay, well, hagfish, Agnatha does not have jaws, but everybody else has jaws. So we're not going to pick something that we can't find continuous through all of it. So that's why you have to kind of look at your chart of what all their traits are and then decide, okay, well, everybody has this or everybody has this, and it could be everybody has this particular protein, but these guys don't. Everybody has this particular protein, these guys don't. So you might be dropping off something like that. And, here, this is how to construct that cladogram. So you look to see, what does everybody have? That's your ancestral trait. And then you just start dropping them off. Okay, take a look at this diagram right here. What one seems like the better choice, on the left or on the right? Which one gets the green light? On the right, okay? Parsimony means, like Occam's razor, is let's do it with the least amount of changes that have to occur, the least amount of things we cannot explain. So more than likely, the red feathers evolved here, and now they have that. They wouldn't have independently evolved red feathers. Probably there was a common ancestor for that, okay? And so you can, let's see, okay, so monophylogeny or monophyletic, that's what you want to get right here, okay? So this is, everybody had this, and then we branched out. We didn't re-evolve some new trait. And I don't think they're going to have you ever, look at this one on polyphyletic, multiple times. You're not going to keep evolving that different trait. It's going to you're going to try to find the most simplest explanation of each of those traits. Okay, and then homologous structures, again, we're looking at linkage, and we already talked about this as evidence for evolution, right? The similarity in anatomy, and even though these are all used, every single one of these is used for different what? Purposes, right? What do we talk about when we're talking about the functions? Analogous structures. Are any of these structures analogous?
So homologous is same function, different
No,
structure?
homologous is same anatomy. They may or may not have a different function.
So, okay. So
Yeah, so these right here, are any of them analogous? No. Do any of them do the same thing? No. This one flies, this one swims, this one walks, this one grabs. So they all have different functions. So I don't care about functionality, but we do care about structure. Because if they have common structure, then they probably have similar DNA, which means they probably have a common ancestor. Yes?
And then vestigial is just having something that was once the same but isn't necessarily relevant anymore.
Right. It was
Okay.
once, it was useful.
Okay.
And so what it does is a vestigial structure points to your ancestral past.
Okay.
Yeah. So that tells you where you could have come from.
Okay.
Yeah. All right. And what's next? Okay, so let's look at the different types of evolution. A is ancestral species in all of these situations. So take a look at this first one. Why don't we have oldest biobuddy explain this one? Divergent. Go ahead.
Divergent is like you have the common ancestor, but you diverge parallel, so you adopt your environment. The parallel is divergent is where you come from two different common ancestors, but you come to make the same. You're in the same environment, so you're like, well, we're the same characteristics. And they literally go, whoa.
So on parallel evolution, we could have a similar ancestor like a small mammal, right? And we ended up with similar structures even though you're a marsupial mammal and I'm a placental mammal, right? And when we look at convergent over here, so we don't have a common ancestor here on convergent, but we ended up in the same way. We both have fins. We both have wings. We don't have, they're analogous. This would be, this type is not pointing to a common ancestor. Yes?
With convergent evolution, could you...
But you could also have it for this too. Okay, go ahead.
But even for parallel two, could you come up with the same conclusion that they're probably living in similar environments which cause them to adapt to the same?
Yeah,
Okay.
absolutely, absolutely. Okay, so then we went through them already, so go. Oh, I did that one. So go. Yeah. So here's exactly what you were just saying. Okay, so we adapted similarly to that environment. Okay, so this is an example of convergent evolution. We already did that. Okay, so I already gave these. Sorry, I didn't look it over. I was just starting to talk. Parallel evolution, I already gave you that example. So we're two types of mammals. We had an ancestral mammal, so we were similar. But, you know, Pangea broke up, so we all went our own ways. But yet, we both kind of look like mice, you know? So that would be an example of parallel evolution. And we did have a common ancestor, though we don't have a recent common ancestor. Okay? And then here, this is looking at classification and taxonomy. So if you look here at the big one, it's Eukarya, right? And then Animalia. Remember how we went through this? Chordata, Mammalia. So in here, we keep dropping people off, right? Doesn't this kind of look like another way to express the same thing about, like when we look at the phylogeny, right? Right? Where we become more and more exclusive. Yes?
Do we have to know the order of like domain, kingdom, phylum?
I would. Domain, kingdom, phylum, right? This is the way do you remember I don't know what mnemonic you use, but I use, did King Philip come over for good soup? Kingdom, phylum, class, order, family, genus, species.
Yeah.
What?
Yeah.
Yeah, no.
Kilo.
Yeah. So did King Philip come over for good soup? Question mark. Hello, Ms. Chevalier.
I'm sorry to interrupt.
It's no
Did
problem.
we get a date for the pigs?
No, I have not gotten that date for that pig.
All right.
As soon as I know, I will give you that date.
Thank you.
You and I will date a pig.
Okay.
All right. I don't know if this question's going to work. Do you want to use it? I don't even know if there are. Do you want to do it or you want me to just keep going? Okay, do it now. Be your favorite shoe. Shoe.
Like brand or like type?
Anything, I don't care. I'm just trying to give you something. I'm in a pause. Okay, we're going to come back to that question just momentarily. We're going to just do the last slide, and then we finished out this enduring understanding. Okay, so genetic recombination, these are all the ways that bacteria can get new traits. Okay, so let's talk about this one. So transduction, what are we using in transduction?
It's like
A virus. Transduction is always a virus. Okay, transduction is when a virus infects bacterium 1 and picks a little bit of host 1 up and then uses it. It doesn't, you know, it just happens in the process because they are really good genetic engineers, that when it goes and infects host 2, it's not only infecting it with its viral DNA, it's also bringing a little bit of host 1's DNA to it as well. That's transduction. Okay? Conjugation, you could think of the word conjugal visits, right? And so in this case, you're exchanging DNA, and in this diagram, they don't show you this, but if there would be a sex pilus. The reason why they have a plasmid there is there's something called the HFR gene, and it's you know how we have males and females biologically? There are males and females if they have the sex factor, which looks like a plasmid. But I haven't taught you that, but that's why they have it in that picture. And then the last one is transformation. And this is when your bacteria pick up DNA from other, like, decomposed or dead DNA or something, or like we're doing it artificially and it's picking it up from its environment. Okay, and that's transformation. And then, see, then we're on to the next enduring understanding. So we kind of close that one out. So I'm going to go back to this question. Did everybody get to go in as their favorite shoe? Okay. I'm going to pause this again. Okay, we're going to finish up. And which of these are not eukaryotes? Bacteria and archaea.
So fungi is.
Yes.
It is.
Yeah. Okay, so eukaryotes, okay, so you have prokaryotes and you have two domains. Domains that are prokaryotes. Bacteria, are you listening to me or you're talking to each other? And your second domain is is archaea, right? So then you have another domain, which is what? Eukaryote. Okay, and underneath that you have protista. Protista. That is generally, not always, there are some multicellular, especially in the algae, but this is a eukaryotic cell, usually unicellular. Protista, you have fungi, and these are kingdoms, all of these. Animalia, right? And what's the other one? Plant. Does that make sense? Okay. And then, which kingdom is mismatched? Fungi is not prokaryotic. And plantae are multicellular only, just like animals are. Yes? Algae is usually under protista now. It used to be like a lower plant, but now they kind of classify it underneath protista.
Okay, so then, okay.
Does that make sense now? Okay.
Cool.
All right. Good job, guys. Another enduring understanding done. Check. Boom. Good job. Yes. I'm just.
This is a lab question.
Oh, okay. Do you want to ask it in case people want to know?
Yeah. So for some of us, I know the K plus and the A plus groups didn't, like, for period 1 at least, didn't run on the gel electrophoresis, but our ligated one did run. Can we still make the same conclusion if we don't have K plus and A plus?
Yeah. If it doesn't look. If you've got different variations on the plasmids, then you must have had different cut fragments. So yeah, you can make an assumption.
Okay, so you can make that.
Yeah.
Okay.
Mm-hmm. Okay. All right. Have a good evening. If you're hungry, have a piece of toast.